Hello and welcome to Basic Medical Sciences. So in this video, we are going to talk about Bacillus species. Right, so the reason why you have this picture is I want to show you the classification. Right, so here firstly, we already covered the cocci, right, all of them. Right, so you can check our playlist and watch all these bacteria. So now we are going to talk about the bacilli, right? The gram-positive bacilli, Bacillus anthracis and Bacillus cereus. That's what we're going to talk about in this video, right? So let's restart. Starting with a Bacillus anthracis, right? So this is a causative agent for anthrax, of course. It is a gram-positive spore-forming non-motile rod, right? Uh, and like on culture, the age of colony shows irregular comma-shaped outgrowths on blood agar, right? So these are referred to as medusa head, right? And I'm going to show you that in just a moment. In some questions, they can refer to Bacillus anthracis as a gram-positive rods arranged in chains, right? So I'm going to show you that again, right? So firstly, I want to show you this medusa head. You can see the outgrowth right here. So this is anthrax on culture, right? So the next one here is showing gram-positive rods arranged in chains. So you can see chains, right? So how is anthrax transmitted to humans? Because usually it's a zoonotic infection. It causes anthrax in herbivores like cattle, etc. Right? So how do we get it? We get it if we get in contact with an infected animal or its products. Right? For example, skin, right, or hide, right? So let's talk about virulence factors, right? So anthrax is actually, you can say it is two groups of virulence factors. The first one is actually a capsule, which is antiphagocytic. Right, so this capsule is actually an exception because it consists of poly-D-glutamate. In general, capsule is polysaccharide, but on anthrax, this is an exception, poly-D-glutamate. Right. So for encapsulated bacteria, we were using this coat, right? So yeah, we have it. The second group of virulence factors is actually anthrax toxins, right? The toxins. So the toxins are actually responsible for local and systemic manifestations of anthrax. All right. So the anthrax toxin is two subunits, right? The B subunit facilitates the entry of the A subunit into the host cell. The A subunit, which get into the cell, is two components: the edema factor or EF and the lethal factor or LF, right? Okay, so let's get into more detail, right? So let's start with the edema factor. The edema factor actually binds to the calcium and calmodulin and gains the activity of adenylate cyclase, right? So if you still remember, adenylate cyclase, it converts ATP into cyclic AMP, right? So in this case, there will be increased cyclic AMP, facilitating the movement of fluid to the extracellular space, causing edema, right? So that's edema factor for you. The second one is the lethal factor, right? This one is actually an exotoxin, which acts as a protease that cleaves MEP kinase. Oh, okay. So MEP kinase is actually a signal transducer. It takes part in cell growth, right? So if it is cleaved, it means there is no cell growth. So what will happen is it will lead to cell death and tissue necrosis, leading to a common sign on the wounds called an scar, right? And I'm going to show you right about now. Here it is. You can see this kind of wound, which is black. It's called an scar, right? Here around. Okay. And here is another image. All right, you can see it again, the eschar. All right. Right, so let's move on to the clinical presentation. All right, so in terms of clinical presentation, we will divide anthrax into about three forms, right? So we have cutaneous anthrax, inhalation anthrax, and gastrointestinal anthrax. What we are going to do is we are going to talk about like each classification, and we will talk about route of entry and the clinical presentation. For example, the route of entry for cutaneous anthrax is actually skin contact, right? And on clinical features, so it's basically a skin lesion, right? The key thing is that the lesion is painless, uh, but it can be pruritic purple also, uh, and it can transform into a vesicle, right? From from pruritic purple to vesicle, and then ulcer with surrounding edema, leading to a necrotic black scar, and it can heal by granulation. At the end, we can have hyperpigmented skin or a scar, right? So this is all about cutaneous anthrax, right? In brief, this is what we do. For inhalation anthrax is, of course, through inhalation, right? And the common manifestation is hemorrhagic mediastinitis, like the bleeding of the mediastinum, right? So this will lead to widening of mediastinum. Like on X-ray, you can actually see it, right? So this form is known as pulmonary anthrax or Wool-Sorter's disease, right? And it's fatal. Mortality is actually 100%. You die, you go. Right, moving to gastrointestinal anthrax. This is mainly through ingestion, right? So if an animal dies of anthrax, please don't eat. Make sure the vet people are there to tell you if the animal is actually infected. Okay. So on manifestations, here are the symptoms, which are of course non-specific, like nausea, vomiting, abdominal pain, severe blood diarrhea, hematemesis, hemorrhagic lymphadenitis, and ascites. Oh, I think now is the time to talk about Bacillus cereus, right? So Bacillus cereus is actually a heat-stable spore-forming bacteria which usually grows on heated food that cools down slowly or improperly refrigerated. Right. The most common, when they talk about Bacillus cereus, they include heated rice, right? So heated rice is the most common, right? And there are two toxins. The first one, enterotoxin 1, is known as the emetic factor, right? So the incubation period is usually 30 minutes to 6 hours after ingestion, and the most common symptoms are nausea and vomiting. The second one, enterotoxin 2, is a diarrheal form, right? And the incubation period is 6 to 15 hours after ingestion, and this will lead to watery diarrhea for 24 to 48 hours. So here is how you can remember heated rice, right? It goes like this: food poisoning by heated rice. Be serious. Be serious. Food poisoning by heated rice. Be serious. Be serious for Bacillus cereus. Please, if you like this video, make sure you subscribe so that you won't miss any of our latest video. Don't forget to check our videos on the following bacteria: Staph aureus, Staph epidermidis, Staph saprophyticus, Viridans streptococci, Group A streptococci, Group B enterococcus, and everything you need to know about virology. We have it here. Just make sure you check our playlist and watch those videos. So that's it for this lesson. Until next time, cheers.